So in the last class, I had introduced you to the lambda phage vectors, the replacement and the insertion vectors, and I had also introduced you to cosmids. And I had told you that the advantage of cosmids over the regular plasmids is its increased cloning capacity. So with respect to cosmid, I told you that what is the way, how do you make a cosmid clone? So in that context, I told you that you need to cut the cosmid vector at the PCS, or sorry, polylinker site, also called MCS, and you have to make your DNA inserts of a particular size because its cloning capacity is around 30 to 40 kb. You can make genomic fragments of that particular size, and then you perform the ligation in such a way that these concatenamers are formed, for which the conditions are generally standardized and mainly requires very high concentration of these fragments. Once these are there, then you have to find a way to get these individual lambda uh, inserts into the phage heads. And once these are made, then you need to, because these are phages, you can infect the E. coli and it will insert the DNA inside the E. coli. Once inside the E. coli means we have been successful in transferring a fragment of around 30 to 40 kb inside a bacterial cell very efficiently. Why very efficiently? Because this infection or transduction process is much more efficient as compared to a regular transformation process. Further, the transformation process doesn't work very good for large size plasmids. So anyway, in this case, the transduction based introduction of these plasmids into the E. coli is very efficient. So coming back to this particular point, that how do you package these into the empty phage heads and make a complete phage particle? So it means that we do have a requirement that we need a phage particle. Yes. Okay. So that we need a phage particle in which the DNA is not there. So how do we get a phage particle in which a pre-assembled DNA is not there and you have the liberty to actually transfer or package your DNA of choice into the phage head? So that process is called as in vitro packaging, and there are two methods by which you can make empty phage heads and particles. So let me just come to those points. So for one of the method, please look on this particular panel. So what they have done is, see, one way to have this pre-assembled, you can just overexpress the proteins which make the phage head and the tail in the lab, purify those proteins and put those together, and then there is a possibility that we do have these pre-assembled pre-heads as well as pre-assembled tails being made. But then the problem is that it is extremely labor intensive, and also it will require a lot of time that you will I don't know, some of you might have worked with purification of proteins, so you can understand how difficult that particular strategy would be. Another option is that you use mutants. So in this case, what they have done, that there is a mutant E. coli in which they have one lambda phage which has a mutation in a particular gene called D gene, and it is staying in E. coli in the form of a lysogen. Means it is integrated into the phage genome. So then you activate these E. coli particles so that it enters into the lytic phase and starts preparing the proteins which are required for phage assembly. So then what will happen, because it has mutation in one particular protein, then rest of the things will be made. For example, in this case, let's say pre-heads including protein E will be made, assembly proteins will be made, but even the tail protein will be made, but the DNA will not be packaged because this particular it was a mutant one. It did not have protein D. If protein D is not there, then the DNA will not be packaged. Similarly, if there is a lack of protein A, so there are different, mutant glycogens which are available. So similarly, if protein A is not there, then it will not package its own DNA. But you can see that it has all the other things which are ready for assembly. So in this case, what they do is, in one of the strategy, that they take the lysate from this particular phage after inducing it from this particular E. coli, and they combine it with, let's say, a purified protein D, and they combine it with concatemerized lambda DNA, the one that you made. So then what will happen, because now all the proteins are there for the assembly of the phage, and they have this concatemer DNA, so these will be then packaged into the phage head, and we'll have a mature phage particle. Another option is that what they do is that rather than purifying another protein and then adding it to the this lysate, they use two different mutants. So in this case, what happens that because the mutation is in the protein E, so we will have this pre-assembled tail. We will have assembly proteins. We do have this protein D also, but because protein is E is not there, so pre-heads will not be made. So if I combine the lysate of this particular bacteria with this particular bacteria, then it means that these two complement each other, and now we have all the things, all the machinery for packaging of the phage particle. And we will provide our DNA from outside, and then we will get a assembled mature phage particle. So these are the few strategies which are used for in vitro packaging. And when you are making, means in many molecular biology experiments, there is a possibility that you do require in vitro packaging. So for example, I made genomic library during my PhD time. So that time in vitro packaging extract mix used to come by the company, and I would just combine my genomic library constructs with the in vitro packaging mix, and it was done. So I didn't need to actually grow these in my lab and harvest the protein. You can just buy it. So okay, so if you remember, I had introduced you to P1 phage, which has comparatively higher cloning capacity as compared to lambda phage. So similar to, for example, they have made cosmids, in which they have, in the plasmid, they have introduced the cos sites and increased the cloning capacity. Similarly, for the P1 phage, they have made pack proteins of P1 based artificial chromosomes, which have much higher capacity as compared to these P1 phage alone. So definitely there are so many cloning vectors. I cannot go in detail with all of these, but this is just for your information. Now coming to M13 phage, and why am I discussing it is not because they have very high cloning capacity. No, they don't have. They have comparatively very low cloning capacity. When I say cloning capacity, means the size of the DNA that can be cloned. But they are required in those cases when you want to harvest the single-stranded DNA for a particular segment of DNA of your interest. Okay. So M13 phage, they are circular and they are single-stranded DNA phages. And if you remember, when I told you that how do you harvest the DNA from various sources, I had also told you about M13 phages. Very briefly, I told you. And so in this case, what happens that this M13 phage only infects the E. coli through the F. pilus. So anyway, so it injects its DNA inside, which is circular, and these then, I'll come directly here. So once it injects its DNA, it then starts making double-stranded DNA, and these forms of DNA are called as replicative form. And so these replicative form, they make many copies of itself, of its M13 phage. And then these are acted upon by a particular protein which makes a nick at the origin of replication of these particular genome. and once it makes a nick here, then these the displaced particle is then cut at the terminal site, and what we get is the circularized single-stranded DNA. This single-stranded DNA is then packaged into the phage head as somewhere near the membrane of the bacteria, and these are then ejected as mature phage particles. So one major difference that you see between the lambda phage and M13 phage is that the extrusion of the phage particles in case of lambda phage required the lysis of the bacteria, but in this case they are just released from the bacteria and bacteria remains unharmed. So in this case, what we do, so this is, let's say, the general life cycle of M13 phage. Now, with respect to your cloning work in M13 phage, what you will do is that you will harvest these replicative forms of M13 phage and work with it just like any other plasmid. So it means that after infection with M13 phage, you can harvest the bacteria. If you harvest the bacteria then and isolate plasmid, you will get these double-stranded forms. I will come to the structure of the cloning vector, but then you can make a cut in these vectors and you can clone your gene of interest. If you clone your gene of interest, it means that now you are ready with a recombinant M13 phage genome. And now let's say if you want to harvest the means so in that case you can easily manipulate it, and if you want to harvest the single stranded DNA form, then you will not harvest the bacteria, you will then harvest the M13 phage that are extruded from these bacteria. So it means in those phages you will have the single stranded form of the DNA of your gene of interest. So why do you want the single-stranded DNA for any purpose? So generally the single-stranded DNA forms are required, let's say, for sequencing purposes, or they are required for site-directed mutagenesis purposes. And M13 phage, they are also required, means they are also widely used for a technique called as phage display method for studying protein-protein interactions. So yeah, so this is about M13 phage and how you can harvest the single-stranded DNA forms of M13 phage. Now the problem with M13 phage, so this is the structure of M13 genome, and as you can see, all the blue region are the essential genes which are present in its genome, and what we are left with is very small segment of DNA. And means what I want to convey to you is that we have very limited capacity of cloning here, because most of the genome is essential in nature, so we are left with very less amount for for it to be packaged inside the M13 phage. So anyway, so one problem with it is that, but the advantage is the single-stranded DNA forms. So for M13 phage, a whole battery of M13 phages, let's say cloning vectors, have been developed, and I'll just briefly go through it. So all of the cloning vectors obviously will have a ORI, because that is required for its propagation. And then they introduce this LAGZ gene here for screening purposes. Then within the LAGZ gene, with a very smart technology, they introduce the equarban site. So this is MP2 vector, this is MP1 vector. See, as the improvements happen, the series number changes. Okay. Then I'll just come to how they introduce the equarban site. But then at the equarban site, now they had the option to introduce the polylinker sequence. So then they had M13 MP7 vector that was developed. Then it was realized that can we have the polylinker sequence in these vectors in such a way that their sequence is exactly same as the polylinker sequence which is present in other vectors. And why do I require that same polylinker sequence should be present in, let's say, 2 or 3 different vectors? Because then it becomes very easy for sub-cloning. Means if I have cloned something here and I want to, let's say, transfer it to a puck vector, which I discussed previously, then it's simply that, let's say it was cloned in BAM PST1. So I'll come cut it with BAM PST1 or let's say SAL PST1, and then I will just transfer it in same orientation in the other vector. So that's why another series M13 MP8, it was developed, in which the restriction enzyme sites are similar in same orientation like in other commercially available vector. So now coming back to that, how did they introduce the equivalent site in this LAGZ prime? And this will be important for maybe your individual experiments that you are doing in the lab, that how you can actually think about introducing a restriction enzyme site without introducing 6 new bases within a particular stretch of DNA. So in this case, when they were carefully looking at the sequence of the LAGZ prime gene, they came across a site GGATTC. Okay, so then they got the idea that if we convert G to A, then it means that we can convert this particular stretch of DNA into the eco-R1 site, because eco-R1 site recognition site is GAATTC. So just by changing one base, they didn't alter much the properties of LAGZ prime. Obviously, you'll have to look into that for your individual experiment, because there is a possibility that if you change one base, maybe it just gets converted to a stop codon, or it leads to some other mutation or some major change in the amino acid which is affecting its folding or its functioning, etc. But in this case, one base mutation that was introduced by site directed mutagenesis, not much changes were made, were happening in the LAGZ prime gene, but means finally they could introduce eco-R1 site in this particular stretch. Okay. So that was the thing about M13 cloning vector. So what you have to do is you have to clone your gene of interest here, and then you can introduce this into the bacteria and harvest the M13 phage, which are in the supernatant section if you pellet it down, and then harvest the single-stranded DNA. But how do we increase its cloning capacity? So in that context, they came across phage-mets. Okay, so phage-mets are means these are the cloning vectors which have origin of replication of the plasmid, and they have origin of replication from M13 phage. So why they were developed is due to the fact, let me come here. See, if you see this figure very carefully, then I had told you that the nick that takes place, which finally leads to these single-stranded forms being developed, it was done at the origin of replication. Okay, so it means that the presence of ORI is very important, and obviously certain enzymes from M13 phage, they are very important for any segment of DNA to be made into single-stranded DNA. So what they did was that in the plasmid, these commercially available plasmids, which have ORI, ampicillin, and maybe a MCS region or a screening marker, etc., they introduced a F1 origin of replication, okay, which was for the M13 phage. So it means if this is there and if the phage rest of the machinery is there, then any DNA, it will be a nick will be made, and then it can be packaged into the phage heads. So how do we, so what you will do is you will just clone your gene of interest in these phagemids. Now, how will you get rest of the machinery? Because this is not complete. There is no other essential gene from the M13 phage. So what they did was that they infected these phages with a mutant phage or a helper phage, which had uh, everything of the phage genome being expressed. All the other genes were there, but it was unable to package its own DNA because its own F1 origin was mutated. So that was a smart strategy. So you will make your clone here, you will transform it into bacteria. Once inside the bacteria, you will transform the bacteria which has the phage with this helper phage. So this helper phage will get inside the bacteria, and all the proteins for M13 phage, they will be made by this helper phage, and they will act upon this F1 origin of replication, and what we will get is the single stranded forms of DNA of our gene of interest. So this is one phage vector that you will come across very often when you're working in the lab, P blue script. So as you can see, there is MCS, there is LAGZ based screening method, there is origin of replication for the plasmid, and there is origin of replication, the F1. Okay, and this is the ampicillin resistance for these plasmids or phagemids to be maintained in the bacteria. Now, the second point is that if you see that there is a plus origin and there is a minus origin. So it means that if you want to get the DNA, the single stranded DNA of one strand or the other strand will depend on which F1 origin you are taking. Okay, because it will happen in that direction only. So you will find some phagemids with a plus sign, and you will also find phagemids available from the company in the minus sign. So, okay, so we have covered these plasmid vectors, phage vectors, and cosmid vectors with much higher cloning capacity. P1 bacteriophage, I've just introduced you to that. And I also told you that some changes have been made in the P1 phage so that it can, accommodate bigger cloning fragments. Now coming to the, and I've also introduced you to the M13 phage, and also that how you can harvest the single stranded DNA from these M13 phage. So when I'm just discussing M13 phage, let me just quickly tell you that these phages, their coat protein is largely made with proteins called P3 and P8. Okay. So what is done is that if your gene of interest, you make it as a fusion with the P3 protein, then what will happen that your gene of interest means in frame fusion. Okay, so your gene of interest will be present on the cover of the M13 phage, that is the outer coat protein. Or if you make it as a fusion clone with P8 protein or P3 protein, any of these. So it will be present on the outside of the M13 phage. So if you want, means I just want to say that the basis of the phage display method is this only, that you display your protein of interest on the outside of the M13 phage. And let's say in a 96 well plate, you have another protein, and you want to find whether these two interact or not. So then you incubate this M13 phage with the other protein, and then you can look for the protein-protein interactions. I just want to say that these are present on the outside, and I think I should have a separate class for that. So, okay. So coming to bacterial artificial chromosomes, another set of vectors. So please, I know that too many vectors are being discussed, but they are all for different purposes, so I cannot skip those. So coming to bacterial artificial chromosomes, they are based on the F factor. The size is around 100 kb, but they have been found to very stably propagate very large DNA pieces of up to 300 kb. So they have excellent cloning capacity, and some of the initial bags had very low copy number because they were based on the F factor of E. coli, which is present in the low copy number. And the genes that are required for back propagation are the genes which are obviously involved in its replication. So we have this, one is like origin of replication. Then we have rep protein, which is also involved in its replication and control of its copy number. And similar is the function of the par gene or the partition genes, which are very, very important for these vectors, for them to be stably maintained, as well as segregated into the progeny of the bacteria. So these are very nicely partitioned into the daughter E. coli cells. Okay. So these are par A, par B, repi, ORES. These are definitely present in these back vectors. Then what we do have is chloramphenicol selection. So in this example, this particular selection is there, but now many different back vectors are available from the market, which have different selections which are present. So this is an antibiotic selection. Then what else we do have is the presence of the sites where you can clone your gene of interest. So these are, for example, in the early vectors, it was just HIN3 and BAMH1. But the new battery of back vectors, they have much bigger options for multiple cloning sites. Then in the same vectors, they have also introduced the two promoters. So if you remember the PGEM series of the vectors I had introduced in my earlier classes. So T7 and SP6 promoters, they ensure that whatever you are cloning here gets transcribed, and if the translation machinery is there in the bacteria that you are cloning into, then that product, that transcript can be further translated. So in those cases where you want the expression products of the genes or the segments that are cloned here, so in those cases you do require T7 and SP6 promoter. And why they are present on either side of the MCS, it depends that if you require the transcription from this side or from this side, you can choose which promoter you want to use in the bacteria. Then another two sites that have been introduced into these vectors are LOXP site and COSIN site, and these sites are the ones which will help in linearization of this particular back. So the problem with so let me tell you that why these had to be introduced separately. If I had to just linearize my back vector, then I could have easily used cut it with BAMH1 site. So if I cut it with BAMH1 site, then it will be linearized. But if I have cloned my DNA into the back vector, which is in the range of several hundred kb, I mean up to 300 kb, then what will happen that the chances of these sites present in multiple region throughout 300 kb is very high. So it's very difficult to find a unique restriction site for these recombinant back vectors. So for that purposes, they have introduced lock sites. So these lock sites, they are recognized by recombinase called Cre recombinase, and a cut is made here. Similarly, COSIN site has also been introduced, which is recognized by an enzyme called terminase, which makes a cut here. So these options are also given here if you want to linearize back vectors. Why in some cases would we want the back vectors to be linearized after cloning? Could be that in some cases I want to get a restriction map of the inserted clone. So if I want to get a restriction map, so what they do is that they linearize from here and then they mark the ends, or let's say they label the ends of back vector from one side, and then they can actually map by cutting it with different restriction enzyme sites and do the restriction mapping of the insert which are present within the back vector. So in general, this is all about back vectors. They are based on the F factor plasmid. What they have done is they have introduced these PAR genes, and what they have also ensured that the genes which are involved in replication, like REP-A and origin of replication, they are there. They have added a selectable marker. They have also added the genes, the MCS. They have also added certain these enzymes which are present on either side of this particular fragment. For if you want to remove this whole cassette, so you can do it in certain cases. If you want to, let's say, remove this whole cassette where your gene has been cloned, then they have also given an option of some rare sites, like these mostly are GC rich cutting enzymes, and they have around 6 to 8 base pair cutting fragments. For example, NOT1, it has a 8 base pair recognition sequence. Now coming to YAC vectors, so its cloning capacity is even bigger. See, up to 2 MB can be cloned in these YAC vectors. So first let me introduce you to YAC vectors, and then I'll make a comparison between the BAC and the YAC vectors. So these are yeast artificial chromosomes. I could have covered these when I am actually, there's a separate class on yeast cloning vectors, but I'm discussing it here because I'm telling you about the series of plasmids with increasing insert sizes. Okay. So the segments which are very, very essential for YAC, let's go through these one by one. One is centromere. So see, this is, let's say, in a eukaryotic system, a chromosome based thing. So of course the presence of centromere is very, very important if you want these YACs to be segregated properly in the progeny yeast cells. So you need these centromeres. Centromere, if you remember, were the sites where spindle fibers they attach, and they ensure that the proper segregation of the chromosomes takes place. Okay? So they do have centromere. They do have ARS, which is, for example, if you remember, the origin of replication for plasmids. So similarly, autonomous replicating sequence is present in YACs. Then what do we have is the telomere ends. The telomere regions are being cloned here because if we want it to be, maintained as a chromosome, and the chromosome, if you remember, its ends are protected by certain sequences called as telomeres. If telomeres are not there, then the cell takes it as a, let's say, a double DNA strand break, and there is a possibility of losing some information from either side of the chromosome. So what we also need is the presence of telomeres. Okay. Apart from these, there are certain other sequences which are present in these YAC vectors. So YAC vectors, they are shuttle vectors. It means that they can be maintained both in bacteria as well as in yeast cells. So if you want any particular plasmid to be maintained in two different hosts, then what you do require is the presence of origin of replication or its maintenance for both the hosts. So if you see that it does have the origin of replication from for the plasmid. So it means that I can maintain my YAC and work with it very comfortably while it is in the plasmid, in the bacteria. And I can also maintain it in yeast cells in the form of a chromosome. So this also we just discussed. What else is there? It has this BLA antibiotic resistance gene for it to be maintained in bacteria. Now, do we have any selection method for it to be, identify the recombinants or identify the yeast cells which have taken up the plasmid? Yes, there are few options which are available, and these are histidine, URA3, and one more is there, tryptophan. Okay, so these three options are there, and they are only based on the fact that you use the yeast host strain which is auxotrophic for a particular amino acid. If it is auxotrophic, so the yeast cells which have not taken up the plasmid, they will die if you do not provide external amino acid. While the yeast strains which have taken up this plasmid, YAC vector, they will survive because they are providing the genes which are involved either in histidine biosynthesis or in pyrimidine biosynthesis, or somewhere here is also for the tryptophan biosynthesis. Okay, so this is how you select the yeast strains which have taken up the plasmids. Now coming to this name called SUP4 gene. So this is little, let me try to explain it. So the yeast host strain that we choose, see this strategy, or let's say this segment, is for the identification of the recombinants, just like if you remember the LAGZ based screening method. Okay, these genes his and ura3 etc, they can tell you that which yeast has taken up the plasmid, but it cannot tell you which yeast has taken up the plasmid which has the recombinant one in which your gene has been inserted. Okay, so for that purpose we need certain screening markers, and SUP4 is one of the screening markers. So what they have done is they have added multiple cloning site here. So now coming back to your introduction to the yeast host strain. So yeast host strain, it has a mutation in a gene called 82. It's a occur mutation, because of which it gives a red color. I'll not go into the whole biosynthetic pathway. I'll just give you an overall scale. Okay. So what happens that yeast host strain will be red in color because it has a mutation in one of the gene. Now, if the yeast has taken up this plasmid in which this SUP4 gene is intact, then what will happen, because this SUP4 gene codes for a suppressor tRNA gene. If you remember, these suppressor tRNA genes are the ones which can complement the mutation which is present elsewhere in the host strain, okay, or in the genome. So because this tRNA itself has a mutation, it will then suppress the mutation which is present in this particular gene and complement it, and what we will get is the restored wild type activity, and the yeast host strains which have taken up the non-recombinants plasmid, they will be white in color. Okay. Now the yeast host strain in which they have taken up the plasmid which has a gene inserted inside this multiple cloning site. So because of that, SUP4 will not be functional and it will not be able to complement it, and we will again have the mutant activity and we will not have the wild type activity, because of which again the recombinants will be red in color. So can we differentiate between the original host strain which did not take up any plasmid, which was red in color, and the yeast host strain which has taken up this recombinant YAC clone in which your gene of interest is there, which also is red in color? Yes, we can differentiate between the two, because the yeast host strain which has not taken up the plasmid and is red in color will not grow in the absence of histidine or ura3 or tryptophan. Okay, so only the yeast host strain which have taken up the recombinants will be red in color. So this was the general introduction. Some written material is here for your this reference. Okay. So now let me tell you that how do you clone in these YAC vectors. So what you do is that you make a cut here. So there was BAMH1 site which was present next to this. So you cut it with BAMH1, and what you get is the left R, okay, which is ending in telomere, and I will get a right arm which is again ending in telomere on one side. So what we have here is a SUP4 gene in middle of which was the cloning site. So here I can ligate my DNA fragments which have been cut with some site which was there, which is present here, and I can do the ligation. So what I will get is my recombinant, or let's say YAC vector, which is linear in nature. And yeah, that is how you make the YAC vectors. Now coming to that, if you see that in the BAC vector, the size of insert is around 300 kb, and in YAC vector, it is around 2 MB. So when they were making human genome library in, let's say, several years back. Then they started making the constructs in yak vector, and they also tried the BAC vectors. But after some time, even though yak has excellent cloning capacity, so obviously because human genome is very big in size, it would have been really great if we maintain our genomic library in yak vectors. But at some point they actually disregarded the yak strains completely and made their representation of human genomic library BAC clones. So why was that? Was due to an observation that the yak clones, they were not they were highly unstable in nature with respect to the fact that lot of recombinations and chimerism was seen in the clones that were present in the clones, in the yak clones. So because of that, see, this is a thing that you definitely don't want, that whatever you clone and feel that it is a representation of the genome of a particular organism, it should not show any recombination once it has been cloned. There should be no chimerism, etc. But it was found that in yak, the clones were highly unstable, and there was, high percentage of recombination that was taking place. So that's why the yaks, they were completely disregarded at that point of time. But does that mean that yak chapter is closed, totally closed in current times, and the preferred vectors are back? Yes, backs were very, very prevalent, and still many of the genomic libraries, they are maintained in backs only. But the scientists have not given up on yaks, and they have found a way to decrease the recombination events. What they have done is that they have used host strains which are recombination negative, and then they are trying to see whether the yaks can now show much more stability as compared to the previous generation of yaks. So if you see some new article on yaks, you will see that a lot of changes are being done in many of the genes which are present in the either in yeast or in the vector itself, to make it much more stable in nature. So again, stressing the point that the field is highly dynamic, and it is only through your basic biology or molecular biology information that you gather by doing experiments and trying to find the function of individual genes which are present in a particular organism. That, for example, now we know that how recombination is regulated in yeast, that we can manipulate those genes and see that if recombination is contained in context of yaks, etc. So yeah, so this was the comparison I wanted. So if you're asked that what is the advantage of BAC over yak, yes, it's the stability. And what's the advantage of yak over BAC? It is the cloning capacity. Okay, so that's all for today. These few slides are part of the genomic library screening. So that will be a separate class where I'll be telling you how genomic libraries are made, and then which are the various strategies by which you screen a genomic library. That will be a separate class. So my next class will be on yeast cloning vectors, some of the early ones. I don't have many points in that, but still I will introduce you to some of the vectors which are required if you want to clone your gene in using yeast as the host strain. Okay, so any questions for today?
Hello, ma'am.